With this episode, we are continuing to explore companies who are working in the gene editing field. This is part one of a two-part video series where we discover why gene editing is at the crossroads of several converging technologies, the who is who in gene editing, where CRISPR came from, and the various CRISPR component delivery methods. The aim of this two-part series is in fact to help you understand what makes Beam Therapeutics so unique in the field of gene editing. And since I couldn't find an easy to understand yet comprehensive overview anywhere on the internet about the companies and various technologies, people involved, well, I made this one. A quick shout out to all of my new subscribers. Welcome. I'm really happy that you're interested in my content, and specifically thanks to Stefan P, who suggested and asked actually that I make a video on this particular topic and company. Although I have a PhD in biomedical engineering and over 15 years of working experience in the healthcare industry, I do not intend to give you investment advice. Please do your own research before making any investments. You are watching Health, Wealth. If you're new to this channel, welcome. If you're not, nice to have you back. The more research I did into what sets Beam Therapeutics apart from all the other companies using CRISPR technology to edit genes, the more I realized that there is no easy to understand and comprehensive overview of CRISPR technologies in order to really understand what makes Beam Therapeutics so unique. I firmly believe that if you want to make informed investment decisions, you have to really understand the industry and the evolution and new technological developments. The challenge with the development of this kind of content to help potential investors make informed and smart investment decisions is to which level of detail I need to go. My goal with this video is of course not to make you proficient to work actually in a gene editing lab and do your own gene edits, but I want to equip you with enough knowledge about the various technologies involved in gene editing and in the industry so that as an investor you are able to make your own informed decisions and wise investment choices. I hope I found the right balance between just enough information in order to help you make informed investment decisions, but not too much information that it is more too difficult to understand. That said, allow me to set the scene why I believe that treatments derived from gene editing technologies are going to be much more prevalent in the future and offer vastly lucrative investment opportunities. We are in fact witnessing today the convergence of multiple different technologies which all play their integral part in the acceleration of gene editing derived treatments. The first of these technologies is gene sequencing. The decoding of the entire human genome was started in 1990 as an international effort, and you may know it as the Human Genome Project. This project was a colossal effort in an attempt to trying to decode the 3 billion base pairs that make up our DNA and mapping those into the various genes that we know in our human body. The Human Genome Project started in 1990, and as a result of this, over many years, we've been able to identify about roughly 20,500 genes, and scientists have been able to identify the location of those genes on our chromosomes and created linkage maps through which inherited traits, including genetic diseases, can now be tracked and better understood. And in fact, throughout the 13-year duration of the Human Genome Project, we have seen an incredible decrease in the cost needed to sequence and decode the 3 billion base pairs that make up the human DNA. Likewise, we have seen an unprecedented acceleration of our ability to actually decode the human DNA. While the decoding of the first human genome took 13 years and cost about $3 billion, we have since then seen a vast acceleration of the time needed as well as the cost decline. In fact, as per the data shared by the Human Genome website, The cost decline we have witnessed throughout the ongoing Human Genome Project and now going into the future is more dramatic than we have seen in the cost decline of electronics, microchips, and computers, which is driven by Moore's law. This is absolutely incredible, and you can be certain that gene sequencing will become part of daily medical practice and routine. And this is a very important aspect in terms of investment opportunities derived from the analysis of the human genome of each individual patient and person. And this brings us to the third of the converging technologies, which is artificial intelligence. You have just learned that the human genome is made up of 3 billion base pairs, that we have identified about 20,500 genes, and you can imagine that trying to understand the relationships between, let's say, environmental factors, lifestyle, your genetic makeup, and disease is a highly complex and multivariable task. But it is a task which is ideally suited for artificial intelligence and deep learning applications. Artificial intelligence operates and thrives best giving vast amounts of good data sets. And with our proven ability to sequence genes, with the more than evident decline in cost needed in order to sequence genes, more and more sequencing data will become available and can be fed into artificial intelligence networks. The more data we have, the better artificial intelligence will be at predicting and identifying connections and links between certain genetic variations, expression of genes, and how these may relate in the context of individual patient lifestyle, perhaps even to the probability of having disease and the manifestation of disease. As a result, we may in the future be able to predict and to better forecast the onset of disease, even the likelihood of a patient developing a particular type of disease, and can devise treatment plans even before disease manifests and patients have a loss of the quality of life. You can absolutely be certain that there are unique and very lucrative investment opportunities in this space. Also, the cost of healthcare is likely going to go down because it is always much less expensive to preventively treat a patient than trying to fix major damage due to chronic or any other type of illness once it has manifested itself. We can also go one step further by trying to combine in artificial intelligence not only genetic data, but other data sources, such as those currently provided and researched by the Project Baseline, for example. I leave links in the description below. This now finally brings me to the fourth converging technology, which is gene editing. The better we understand what type of genetic variations may lead to disease, the better we are at identifying the locations and the actual targets for specific gene editing applications. With the ability to edit genetic code, we finally have the tools available in order to try and alleviate or correct basically genetic disease altogether. With gene editing, we finally have the tools in order to modify our genetic code and try to alleviate the symptoms and manifestation of not only genetic but perhaps any other type of disease in the future. As you are undoubtedly aware, many companies are already working in this space and some of them already have ongoing clinical trials. This then takes us full circle and we have identified the four converging technologies that will help accelerate the treatment of genetic disease. Tissue biopsies provide the genetic information combined with bioinformatics for the analysis of that information can lead to the development of personalized treatments. And as we increasingly see in the treatment of cancers, may lead to so-called living therapies, where genetically modified cells are reintroduced into the body in order to help, for instance, fight cancer through the action of the own immune system, rather than relying on the traditional treatments of cancer or against cancer, which have not been as successful as we would hope. So let's now look at the CRISPR-Cas system for gene editing purposes and learn a little bit more about these types of applications so that then we are well equipped in order to better understand how the technology developed by Beam Therapeutics is different and perhaps even better. The acronym CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats, is a mechanism that bacteria have developed and evolved in order to fend off viral infections. You may be surprised that not only we are subject to viral infections, which of course is a very current topic given the COVID pandemic, but also bacteria are under viral attack and had to evolve defense mechanisms. Viruses which are capable of infecting bacteria are called bacteriophages. As a quick side note, prior to the omnipresent use of antibiotics, bacteriophages were an area of very active research in an attempt to try and cure bacterial infections. Interestingly, this takes me back to my graduate days when I was working on my PhD in biomedical engineering. I took a microscopy class, and in one of the assignments, we were allowed to use an electron microscope, where I was able to capture this image of a bacteriophage head and tail at 495,000x magnification. So you can see these things are extremely small. Though, back to gene editing. The various cells in our human body is organized into tissues, and as such the cells specialize into certain specific tasks. You can view those cells as essentially microscopic 3D printers who run on genetic code in order to provide all the building blocks that are necessary for life, such as specific proteins. If there's an error in the code, the proteins that are being produced are going to be different and perhaps faulty and cannot perform the vital functions that are necessary for the thriving of the cells and the organs and ultimately the entire human being or other organism. Traditionally, we have been able to modify genetic code, although in a very slow and tedious process, through the process of specific breeding. And this has been, of course, applied to livestock, crops, pets, for instance, dogs, and so on, and this essentially has been an effort of trial and error. The long duration, of course, always necessary because you need for one generation basically to become sexually mature and then you reproduce again and you see basically the traits in the offspring. So let's now focus on the who is who in the field of gene editing. And I don't mean in terms of the various companies, I mean of the actual scientists and people who have developed and work in this particular field. I believe that as savvy investors, you need to know who has actually founded companies, who as a scientific advisor, who is still working in the company in order to give them scientific guidance and expertise. So, without further ado, meet Emmanuel Charpentier and Jennifer Doudna. In 2012, they published a hallmark paper describing the CRISPR-Cas9 system they engineered to controllably edit genes. And it is for this work that they received in 2020 the Nobel Prize. Only 1 year later after their initial paper on the CRISPR-Cas system, Jennifer Doudna published a paper in 2013 with the first evidence for gene editing in animal cells. However, just 4 weeks prior to the publication of Jennifer Doudna's paper in 2013, Feng Zhang from MIT published a paper 4 weeks earlier demonstrating the editing of genes in human cells. The inventions described in these papers, as well as the timing of the publication of these papers, are the origin of ongoing patent disputes. And I am sure in the future we will hear more on those patent disputes. So, now that we are familiar with the origin of the CRISPR-Cas system for gene editing and the inventors behind this technology, I would like to switch gears a little bit and focus on some of the technical challenges surrounding the delivery of CRISPR components into cells. The key problem is that the CRISPR-Cas complex is rather large and cells don't let anything that's really large easily pass through the cell membrane and enter into the cell. This is why there is continuous research into the safe and efficient ways for the delivery of therapeutic CRISPR components. You probably guessed it, but successful methods for the delivery of CRISPR components must meet multiple requirements. To name a few, these are the absorption characteristics, distribution within tissues and cells, metabolism, the overall excretion, and disease characteristics. The ability to deliver CRISPR-Cas components to the right cells in the right amounts is paramount because otherwise regulatory approval would never been granted. And patients expect and deserve efficacious and safe treatments. After all, this is the only way that we as investors are bound to make money. Briefly explained, the delivery of CRISPR components can be divided into two different categories, and these are viral vector delivery and non-viral delivery methods. Let's focus briefly on viral vector delivery. Because viruses are very well attuned and adapt to delivering foreign genetic material into cells, viral vector delivery is oftentimes used in vivo applications. The advantages are that there are many different viral vectors available, generally with a high titer, and low immunogenicity, although the latter is still somewhat debated. There are also disadvantages to be mentioned. Viral vector delivery offers relatively little control over the integration site. There are some publications mentioning the risk of insertion near genes which may control the cell growth and therefore could pose a potential cancer risk. One example of viral vector delivery for a CRISPR-Cas system was the Editas and Allergan study with their candidate EDIT-101 and was the first in vivo use in an adult human. Non-viral delivery methods are often used in immuno-oncology. They offer the following advantages, such as greater control of the lifetime in the cells, reduction of off-target insertions, as well as a reduction of toxicity. And the non-viral delivery methods can be further divided into two subgroups. Those are the physically mediated carrier methods, as well as the chemically mediated carrier methods. Amongst the physically mediated carrier methods is essentially electroporation, the most common use in order to use a physical force which drives CRISPR components across the cell membrane and into the cell. Similar methods are microinjection, magnetofection, as well as hydrodynamic injection. As mentioned, these approaches are often used in ex vivo applications, such as for the creation of CAR T-cell therapy. Amongst the chemically mediated carrier methods are zeolithic imioaerosol frameworks, metal-based nanoparticles, as well as lipid-based nanoparticles. The latter seem to be frequently used as they offer high delivery efficiency, low cytotoxicity, as well as low immunogenicity. I'd like to quickly focus on one example of the disadvantages of viral vector delivery in the lack of control over the integration site. Concretely, I came across this in one of my recent videos on Bluebird Bio. Bluebird Bio use lentiviral vector for the delivery of their CRISPR-Cas system and recently had to investigate two serious unexpected adverse reactions to investigate. If you'd like more information on Bluebird Bio and the recent event, I'll leave a link here as well as in the description below. I have highlighted the relevant section of their press release in yellow, and quite frankly, I was a little bit shocked to actually realize that Bluebird Bio didn't really seem to be all that certain where the insertion of their lentiviral vector actually occurred. It is my understanding, but perhaps I am wrong, that it is only through the additional scientific assessments as part of their halted clinical trial investigations to find out where exactly the insertion had occurred. So perhaps they were lucky, I don't know, but it seems that the insertion did not occur in a region of the gene that is regulating abnormal growth or cancerous behavior. This, however, really clearly highlights the need for precise delivery methods where you know where any gene editing actually occurs. So you need specificity really for the target site. This wraps up part one of our two-part video series on gene editing, different technologies. that will prepare you to better understand what Beam Therapeutics is all about. Make sure to subscribe to my channel so that you don't miss my second video where I go into the details of what makes Beam Therapeutics so unique.